Hello, today we're going to explain protein synthesis. So, protein synthesis consists of two steps, transcription, then translation. Transcription involves the conversion of gene to mRNA. Translation involves the change of mRNA to a protein, in which mRNA is translated. So, to remember transcription with the C, so it's like copy, where a copy of the gene is formed, which is the mRNA. Then we must understand what is codon. Here we have the genetic code, which is a triplet code because it's made of 3 bases. And this genetic code is found in the DNA. However, the codon is just the same with 3 bases. However, it's found in the mRNA. Each codon codes for specific amino acids. There are several properties for codon in which it is degenerate, universal, and doesn't overlap. Another important thing I want to tell you is that in mRNA, A binds to U, not T. Degenerate, nearly each amino acid has more than one triplet code or codon to reduce risk of mutation. Universal, meaning that we have the same amino acid. If we have the same amino acid, it's going to be the same triplet code or same codon for all living organisms. Another thing is that they don't overlap. 3 bases are red, then the next 3. It's not like this way, as I highlighted in red, where in the first step in transcription, gene unwinds. The gene unwinds where the hydrogen bonds are broken by RNA polymerase. And let's go back to see that RNA polymerase is the joker, meaning that it does everything in transcription. Then we have that strands for transcription, where the strand that is above is going to be the template encoding. It's template because, as you can see, the mRNA formed is going to be just identical to it, except that T is U in the RNA. Anyways, it is identical, as well as non-coding. Non-coding, or the characteristic of coding, means that they pair up according to a complementary base pairing rule, which is A binds to U and C binds to G. The other strand, which is below, it's coding because it's complementary to the mRNA, and non-template because they're not identical. mRNA is formed also by RNA polymerase, where it joins free nucleotides by phosphodiester bonds. Then the mRNA accesses the nucleus through the nuclear pores in the nuclear envelope to reach the ribosomes in the cytoplasm. Then translation begins, where ribosomes bind to the mRNA. tRNA anticodon. tRNA is made of 2 parts, anticodon and the site of attachment that is specific to a specific amino acid. So the anticodon pairs up with the codon on the mRNA according to the complementary base pairing rule. Then you have a polypeptide formation where tRNA is attached to a specific amino acid. Then you have a polypeptide formation where tRNA is attached to a specific amino acid. 2 tRNA bind to the mRNA at a time in the ribosomes. There are 2 amino acids because we know that tRNA are attached to amino acids, right? So amino acid with amino acid are joined during condensation reaction by peptidyl transferase to form, guess what, peptide bond. Just the same, peptide, peptidyl, like no difference. The transcription stops when stop codon is reached. AA means that you're annoying, UGA, you are gone, UAG, you go away. Then the protein will enter the rough endoplasmic reticulum for modification and folding. Here is a comparison between mRNA and tRNA. Imagine that mRNA is a messenger that sends the design from the office, from the nucleus, or the genetic material, to the construction site, which is the ribosome, where the truck, which is the tRNA, will contain the small bricks, the basic components of your building. These bricks are the amino acids, and your building is the protein, where these amino acids are going to be arranged according to what's written in the design, what's in the genetic material. The genetic material guides the amino acids to be arranged in a specific shape and structure.